Belief 5, Claire. A new variant spells doom.
Right. Yeah.
Tell us about variants, first of all, maybe. Is this unique to the COVID virus?
So, yeah, okay. So with influenza, there were changes in the virus over time. And if you go back to 1968 and 1957, the public health officials were really scared because there was this big shift in the genetics of influenza, which meant that it became a novel virus. Yeah. But novel meaning some of it was new, not all of it. There's a little part of it looked new to the human immune system, but the human immune system does, coped with it because it, as we said last time, it's got a load of different strategies. Anyway, the public health officials were worried, and along comes this new strain of influenza, and then passes through the population, and then it comes back the following year as the equivalent of a variant, and then passes through a few more, and then so on and so on until it's worked its way through. And so what's happened in the meantime, of course, is that viruses are constantly mutating. So whether or not it's the viral genetics that's allowed it to surge, that's the kind of official view, is the virus mutates. It gets to a point where it can evade the immune system, and it's now primed for another attack, and off goes another wave. But in reality, the virus is mutating all of the time. And the variants that we've seen cause spikes often have been around for many, many months, not causing any kind of a surge. So, we know that already. So I think the surging is to do with all sorts of other factors that we talked about last time.
Seasonality.
Exactly. And if it's not the season, then how come it's always peak in January? If it's just the genetics, it wouldn't always be peak in January. So there's other things causing the surge. And then whatever the virus looks like at that time becomes the new variant, because it will have changed since the last time when a surge occurs. And there was actually, with Omicron, there were 2 waves of BA1, if you remember. So that kind of confirms that theory, that BA1 was still the one knocking about, but there were 2 separate surges because it was other things that were causing the surges and the falls. So I think the variant story was really, really hyped up. Scariants, they were called, weren't they? Scariants.
Yeah.
And I think that's
is it true that a new variant could increase transmissibility and pathogenicity? Is that?
So that was always the story. Every single new mutation was going to be more deadly and more transmissible, and let's all panic. Whereas in reality, the differences between one variant and another have to be small. And they have to be small because a virus, they only have a certain number of proteins, and these proteins have to function. And so if you have mutations that cause a big change in shape to a protein, that protein won't work properly anymore, and that virus won't go anywhere. So the ones that are actually managing to do anything can only cause tiny, tiny little changes so that the virus can still function. The idea that it can be very different to anything that went before is just, it never can be. It can never be very different. But what people got very distracted by was the fact that each one did seem to be more transmissible. Now, this was quite interesting, and it was Will Jones that really kind of got to the bottom of this. That if you looked at the percentage of people in a household that contracted the infection, like we talked about before, that number was higher at the beginning of a wave, and it fell through the course of a wave, and at the end of the wave it was lower. And then the next one comes along and it does exactly the same transition. But of course, if you're measuring it at the point in time, you're seeing a high one at the beginning of a wave for the new one, and a low measure at the end of the wave for the old one, and you can always make the argument that it's more transmissible, even though it's starting at exactly the same point that the previous one started.
Yeah. So was Omicron more transmissible?
Right, well, that's a great question, and I don't think it was. So that first Omicron, the first Omicron started off with that same 10%, right? This is the only Omicron wave they've ever given us that number on, but it was the same 10% overall of the household were susceptible. So if you've got the same number of people who are susceptible, then what we saw was a much more sudden surge, right? And so it was sort of, that makes it look like it was more transmissible. But what we also know is that the period between contacts and infections went right down to
Shorter,
3 days.
yeah.
And so once you've got a much, much shorter period for people developing infections, that will cause a much more rapid surge. But the point is that the size of the spike is basically the same. The number of people affected isn't loads more, it just happens quicker because it just happened quicker. So yeah, it was different, but I don't think it was way more transmissible. One of the other big differences with Omicron is that it stayed much more in the upper airways. It was more cold than a lung infection.
For sure. Was there any stage in the pandemic where a virus became more deadly? I mean, was Delta more deadly than the original Wuhan strain, or was it more deadly than the Alpha strain, or was the Alpha strain more deadly than the Wuhan strain?
So I find this much, much harder to unpick, actually. And I think, again, if you just go back to first principles and say, okay, so you're suggesting that you've mutated the virus a little bit and changed the size of the proteins a little bit, and then loads of people died more. It's like, well, what about that bit? What about that last bit of the argument? How are you going from a slightly different protein sequence to lots more people dying? What was it about that protein sequence, that protein shape, that caused more people to die? And I just don't quite get that last bit of the puzzle. You know, the ways people died of this were the same. And I don't think that a little mutation here and there in the protein has massively changed how people died. They died the same way as they ever did. I do think there was a problem with Alpha, but I don't think the problem was entirely the virus. So if you look at the proportion of people in nursing homes in America who died per case, then you've got a problem early on when they weren't testing. But if you just sort of look from summer 2020, when the testing was up and running in nursing homes and they were doing it quite thoroughly, then what you see is when the vaccine rollout happened, you've got a much higher deaths per case in that period. And what was interesting about that is they did it quite late in America. So they'd had their peak deaths in January, but the peak deaths per case in nursing homes was in February of 2021. And then it falls right down much, much later. So there was something going on there, I think, when we were injecting people who were already quite vulnerable, and their immune systems became very occupied with dealing with all of this spike being made around their body. Then when their immune systems are occupied, it can't be fighting off, doing the day job. And so what we also know actually in that period just after injection is that people are more susceptible to other viruses, or other viruses that were dormant reactivate, and that the mortality rate from COVID was higher generally in the population as well. So there is a period just after vaccination when people are much, much more vulnerable to catching and dying of COVID. And that's, you see that in the data from lots of different angles. So I think that distorted the view about the Alpha variant. People were looking at that and saying, oh, it's a more deadly virus. Well, I don't think it was a more deadly virus.
Interesting. And the immune system, the natural innate immune system, the acquired immune system, is absolutely wonderful and brilliant, but it's not a great multitasker, is it? Well, it kind of likes to do one thing at a time.
Yeah, it has a capacity. And so what you see is that people who've been injected with the Pfizer, this is Pfizer's own data, the lymphocytes, which are one of the types of white blood cell that circulate in your blood, and the blood levels of these lymphocytes went through the floor for a few days. And then it starts to recover. They sort of measure it at day 3, and you're like, well, how low did it go? Because by day 3, you might be back on the recovery path. But those lymphocytes, were in tissues probably. So they're not in the blood anymore. They're in the tissues dealing with all of this spike production.
Right. So there's a lymphopenia after the Pfizer vaccine, a reduction in the number of lymphocytes, because the lymphocytes were busy trying to beat up all this protein that was being produced, which we now know is all over the body.
Yeah, and the thing is, that was the entire design of the so-called vaccine. The design was to mimic the virus, cause a cell to produce a foreign protein. The immune system will then see that cell as foreign and kill it, and then the immune system would learn how to do that for if it happened in real life. There were aspects of that went terribly wrong. Like, it was sold as staying in the arm and staying in the muscle, and of course it didn't. And so there are cells all around the body that were being killed by this mechanism, by this very design.
Yeah. So cells all around the body will be producing this spike protein, and the the cytotoxic T cells would be basically eradicating those cells, killing your own body cells.
Yes, and if they did that in the arm, and if it was a small number of cells,
and you've got a sore arm, so it's no big deal really.
Yeah, and the end result was your immune system was educated in a way that was helpful, then all of that would not have been so bad. But each of those parts were problematic. That's the problem. Each of those things went wrong.
If you're making spike protein in the vascular endothelium of the coronary arteries, it's a rather different prospect to making it in a...
Yeah, and I think, that's the key point, isn't it? That we know that we do know that the lipid nanoparticles went all around the body. They were designed to, because they were designed as a way of having gene therapy. So if you've got children who are missing a gene, that gene's missing from all around the body. So you want lipid nanoparticles to get the gene everywhere.
It's so not what we were told, though, isn't it? It's just...
Well, no, but it is what they knew. They knew that's what lipid nanoparticles were designed to do. But it was not what we were told, absolutely.
Yeah, move on from that, because I'll just get cross.
Or you'll get banned.
Yeah, just so... I'm just outraged, totally outraged. So you mentioned in this chapter, Claire, that viruses don't really want to kill people. What did you mean by that?
Okay, so a virus, and actually just before we go on to this, just a little touch on...
Please.
We've heard lots of people saying there's no such thing as the virus, right? The virus is a lie. And I think it's a really important debate to be had. So I'll just touch on that.
Please.
Because, I do think it's important to debate anything. And we have, as biological beings, a replication system. All of our cells are born from another cell, and our nucleic acids replicate. When you've got a system like that, you will have viruses. That just is what happens in replication systems. When you've got code being replicated, it's why we have computer viruses. So, in principle, it would be very strange not to have viruses around. And what we understand about viruses is incomplete, right? As all of science is. There are missing parts of the picture, but there's all sorts of aspects of the picture that we do understand, and that are evidence for support of a virus model. So, people who share the same environment become ill at the same time. And we have evidence of this RNA sequence that these people are way more likely to test positive for than people who are otherwise healthy. And then we have evidence of the protein that RNA produces that matches up with that sequence that you can detect using antibody testing, which is what you have on those LFT tests, the plastic ones people use at home. And then you have those same people developing antibodies to that protein over the course of a few days, which remain in their blood. And then you can look at, there's all sorts of little pieces of information which go towards supporting that model. And it might be that model is in some way flawed. But until people can produce another model that explains it all, then it's just pointless. Like arguing about this isn't perfect evidence. It might not be perfect evidence, but without another model of how the world works, we're sticking with this model.
It does make sense because we know the sequences of the RNA bases. We know that those three of those sequences, it's called a codon, isn't it? Three of those sequences code for a particular amino acid. We do get these strings in a particular order. And the thing that convinced me the virus exists, we've got photographs of it. The electron micrographs seem pretty convincing to me.
Yeah. Some people are not convinced by the electron micrographs. They think that those are just exosomes that are just, I think
we're onto fringe material there, really.
We really are. And there's also the crystallography studies where they're actually looking at the shape of the receptors and how they match to the ACE2 receptor in a human. And once you're on that level of fine detail, I think that...
Yeah. I'm personally satisfied that Robert Koch's postulates are met.
So Koch's postulates are the bacterial ones. So it's rivers for viruses.
Is it?
Yeah. Only because the virus cannot be on its own, cannot be replicated on its own. So you've always got to have.
That's interesting because I kind of thought it was Koch's postulates because you can duplicate the virus in a appropriate cellular medium. Yeah, but with the bacteria, you don't need another cell to do that. No. So that's the difference, really. But and that work's been done on hamsters and on they have gone through and done that work. So anyway, so we can agree on the virus as an idea.